So for today's advanced concepts presentation, I will be talking about how heme and oxygen bind and the importance of that in oxygen transport and release in the body. So first, I will answer how oxygen binds to heme. Then I will discuss whether any structural changes occur when that binding does happen. After that, I will discuss the differences between the binding capacities of oxygen and heme in the lungs versus the capillaries and tissue. Last, I will discuss how carbon dioxide and water enhance oxygen transport efficiency, and I'll link that to the Bohr effect, which is a regulatory effect of oxygen binding with the interaction of carbon dioxide and hydrogen ions. So first, it's important to know what heme is, where it's found, and what its structure is. So heme and heme groups are found in hemoglobin and myoglobin, and are responsible for the red color as well as being responsible for oxygen binding. So a heme group has an organic component called a protoporphyrin. This protoporphyrin consists of 4 chiral rings that is highlighted in red. So 1, 2, 3, and 4 chiral rings. These chiral rings are linked by a methane bridge, which forms a tetrachiral ring. Surrounding the chiral rings are 4 methyl groups, 1, 2, 3, and 4. 2 vinyl groups, 1 and 2, and 2 propionate groups, 1 and 2. Then we find the key player in oxygen binding, which is the central iron atom. This central iron atom is responsible for oxygen binding. And it's important to understand structurally what is happening with this iron atom. So let's say that it's a 3D molecule, which it is, and the green iron that is facing you or me right now has 2 opposite sides. One side is occupied, and that side is the 5th coordination site. At this 5th coordination site, there is an occupation by the imidazole group of the histidine. So the fact that this side is occupied means that oxygen will not be binding there. However, we have the other side, the opposite side of that, with the 6th coordination group. And this 6th coordination group is actually unoccupied, so that is where oxygen will bind. One reason that the iron has these 2 spaces or has the capacity to bind more is because it is in the ferrous oxidation group, or it has a 2+ charge. It has not been fully occupied to have a 3+ charge. So when the oxygen does bind to the 6th coordination site, there is a structural change, which we will see in the next slide. So the structural change occurs, and we should state that, or I will state that before the molecule is oxygenated, the iron is actually out of the plane of the porphyrin ring. So that is due to it being too large to actually fit perfectly in the porphyrin ring. So in this animation, we see that the heme is actually domed. It creates a dome because the iron is non-planar. But when the iron becomes oxygenated, it becomes planar and flattens. And this is due to a slight or a small exchange of electrons between the iron and the oxygen that effectively makes the iron smaller than it was before. So now it fits into the porphyrin ring. Next, we can talk about the binding capacities of oxygen and heme in the lungs versus the tissues. I'm realizing that the O2 has a uppercase 2 and not a subscript, but I just want to acknowledge that I know that should be the subscript. So, getting back to the presentation, the lungs have a higher, in the lungs, heme has a higher affinity for oxygen than in the tissues. This is due to pH, which we'll see has an effect on oxygen release. So the pH in the lungs is about 7.4, and the partial pressure of oxygen is higher in the lungs at 100 torr versus the tissues where the pH is 7.2, so lower than that of the lungs, and the partial pressure of oxygen is lower in the capillaries at 20 torr. There's even a signal molecule that's released by active muscle that decreases the affinity of hemoglobin for oxygen. So here we see that the binding capacity of oxygen and heme in the tissues is lower than that of the lungs. Next, we find that this difference in pH and partial pressure actually is responsible for a 77% carrying capacity of oxygen. So that difference between the 7.4 pH and the 7.2 pH of the tissue causes there to be a higher amount of oxygen carried. I should also state that that value of 77% is higher than if there were no pH change. If there's no pH change, about 66% of oxygen will be carried. Then we can move into the Bohr effect. The Bohr effect is actually the effect that hydrogen ions and carbon dioxide interactions have on oxygen release. When carbon dioxide and water interact, the pH drops. When this pH drops, we see that the saturation of hemoglobin is lowered. So, in the presence of carbon dioxide, hemoglobin's oxygen affinity decreases. So we're going to have a release of oxygen. So we can liken that to tissues that are far away from the lungs or that take longer to get oxygenated blood to. If they have a lot of CO2 in them, the body's going to want to oxygenate them for proper functioning. So one way this occurs is that the carbon dioxide reacts with the terminal amino group of the deoxyhemoglobin and creates carbamate. Carbamate is negatively charged, and this stabilizes the deoxyhemoglobin and actually favors oxygen release. So that is how oxygen will be further released, and that is the effect of pH on the capillaries is that oxygen will be released there and that there's a lower affinity for oxygen when there is carbon dioxide present. Thank you for listening.